In this video today, we're going to go over how to run an SDS Page protein denaturing gel, and we're using that on an expression system so that we can make sure that the protein that we're trying to express in our cells is actually working. So to start off, we need to go to our cell culture, and we're going to take a sample out of our cell culture. So I take 100 microliters out of my culture bottle, and then I go ahead and spin that down at the max speed for about 3 minutes. Once that's done, I remove the sapronatant and just look for the cell pellet, and I resuspend that in 40 microliters of 2x SDS loading dye. While that's going, what we're going to do is set up our gel. So I put some parafilm on the bottom just to prevent any leaking from happening, and then we're going to go ahead and make our gel solution. So we're going to have a 15% acrylamide on the bottom of the gel, and we're going to have a 4% acrylamide at the top. So this is going to allow all the proteins to run together and be able to separate at the same places. So we're going ahead and adding our different solutions to our mix. And then once we add our TMed, we're going to go ahead and pour that into the gel. And then I put a little bit of ethanol on top of it just to prevent it from drying out. And then we can pour that off once it's fully polymerized. After that is done, we're going to add the 4% acrylamide on top of that and then insert the comb and let it completely polymerize. So while that's going on, we're going to go ahead and boil our samples. So I boil these at 95 degrees C for about 3 minutes, just so that it goes into solution. And then if it sputters at all, you can spin these down, but we can go ahead and load these now. So what you're seeing here is I'm going to load 3 microliters of my protein ladder, and then I'm going to add 10 microliters of each of my different samples to the wells. Once all the samples are loaded, we're going to put this back in the box and go ahead and start this up. So I started at 80 volts for about 14 minutes, which allows all the proteins to line up, and then I switch it over to 180 volts and allow it to continue to run. So since we put the ladder in there, we're able to see where our proteins are going to be spaced out. So I use the ladder as a reference to see how long I need to run the gel for. Once it's done running, we're going to go ahead and take out the gel. So you got to be pretty careful in removing the gel. I separate it from the sides and then gently pick it off before we add Kumasi stain. So this is going to allow the different proteins that we have to stain blue, and therefore we can see them. So after that's gone for about an hour, we pour the Kumasi back, wash it off with water, and then we're going to add our de-stain solution. So when I do this, I actually add some Chem wipes to it just so that it can soak up some of the dye. But after that's gone for about an hour, we can look at it on a lamp and see what the gel looks like. So you can look for your protein bands and make sure that everything looked the way that you expect it to. The longer that you let your gel sit in the de-stain, the prettier it'll look and the more clear your proteins will be. So that's everything that you have to look at for running a expression gel.